Hello everyone, I'm Khurshid, and in this video I'm going to explain the paper called Visualizing and Understanding Convolutional Networks by Matthew Zaylor and Rob Fergus, which was published in 2013. I think this is one of the important papers for understanding CNNs, the short name for Convolutional Neural Networks, and this paper basically explored how we can visualize filters in the convnets and how these filters indeed represent hierarchical concepts that we learn from the dataset. Okay, at the beginning of this convolutional neural networks era, we knew that most of the operations in convnets were convnet filtering layers, ReLU operations, and max pooling operations. So we had certain combinations of them. Zaylor and their co-workers, basically co-authors, basically offered these reverse operations for this every operation. Basically, after the every operation, we have an activation map, so to say. After convolutional network, we have this activation map of filters. After ReLU, we have rectified activation maps. After pooling, we have pooled activation maps. So knowing the activation map at the end or at certain point, we can basically reconstruct the input for the last operation that produced this activation map. From the image, you can see that if we know the activation map here, we can basically apply the undo operation on the every process we have. For example, for max pool, we have unpool. Then we can reconstruct the activation map in between ReLU and unpool. And then the undo operation for ReLU is basically, again, ReLU. We can apply that ReLU to that activation map we produced to come up with the activation map in between convnet and ReLU. And then they've also defined a convnet, that is a deconvnet, to undo the convnet, which is basically using the filters and biases of the convnet, but in a different way to undo this particular convnet operation. And then having these learnable filters that are basically learned during the training phase of the convnet we have, that doesn't require any training in this deconvnet part. We can basically visualize filters, either filters themselves or the receptive field of these neurons that are activated on the image basically we give to the neural network. Let's see how it's done. So to say, I should here mention that sometimes we have several kind of convolutional blocks, each of which contains several operations. Since the operations are sequential, we can basically can undo everything on the way without any kind of problems. So knowing these operations defined and having these operations defined, authors claim that or concluded that indeed filters in the first layers represent simpler objects such as edges and colors, and then their combinations indeed represent whatever we detected in the layer 2. So layer 2 are basically compositions of filters or the patterns in the layer 1. And as you can see in layer 3, we have the compositions of filters in layer 2. And as we go deeper, we can see more interesting concepts such as texts, human faces, and then I can see even typing machine, and dog faces, and so on. Okay, how it's done in code. To run this exercise, I prepared some code. One is util.spy, which contains basically auxiliary functions for visualizing and then preparing data, which do not have like high importance. I just hide them in the code to not to have lots of code. And I also prepared this notebook, which I'm going to share with you, that basically helps with downloading of the dataset that I'm going to use, and the dataset class, and the convnet I'm going to use. The dataset itself is called ETHEC, ETH entomological collection. Basically, hierarchical classifications of butterflies like this. So they have families, subfamilies, genus, and species. I'm just using the family labels as the classification target. So I have 6 families, and I have images of butterflies, and we need to write a neural network that basically classifies these butterflies into their own families. Really interesting data set, and this is really a good data set for learning convnets. Okay, so in VGGNet, I have this class called ithack dataset that basically reads the dataset from the downloaded folder. So I have this kind of code for downloading and extracting already also. And this is like a PyTorch dataset class that you've seen already in different places. And I'm using CUDA as the GPU. And I'm also using VGGNet class, VGG8 basically, with 8 layers, and it has 4 blocks, and every block contains 2 convnets for the first 3 blocks, and the fourth block is a little bit simpler, and every block contains 2 convnets, 1 pooling, and and in between we have ReLUs defined in the forward function here, as you can see. We have convnet, ReLU, pooling, and so on. For this NN module class, VGG convnet, I'm also defining a function that basically returns the patches and the receptive field of a certain filter, of all of the filters in certain layer. And then I also have obviously I don't need to return all of the patches because they're really kind of huge, squared order basically on the image. So instead I'm returning some kind of certain number of patches from the image. I'm defining here the operations that we need to perform on the input. So these are the operations that we are indeed using here. I'm not adding linear ones because we are not interested in the visualization of linear layers here. So I'm basically performing every operations in this ops function, ops list, until I basically reach the case convnet in the list. There is no need to continue processing the input because now we are not interested in any layer after these convnets. So knowing that filters of that layer and then their activations here, we can basically get here the number of patches is 9, so 9 best activations for every filter, and their positions as indices here. We also can get the max value activation for every filter on that particular layer, so that we can sort the filters in this particular order. This would help because we want to visualize only the filters that are basically detecting something. So it might be possible that, for example, a filter that is responsible for detecting a human head might not be firing anything because we gave an image of a car, so to say. So there is no need to visualize that. After performing the operations on the input, now we need to perform the reverse operation on every operations we've done so far in this reverse order of the operations. How this is done? So basically, for every filter and for every activated neuron, the best activated 9 neurons, I will basically get a 0 activation map. So activation map which has the same shape as the output of the case convnet. And I put that neuron that was fired with high value to its kind of value. So everything becomes 0, only that neuron becomes activated. So if I start performing these operations like backwards, then it should be the case that the pixels of the input image kind of computed from that neuron. In other words, I'm asking the question of which pixels of the given image should be fired to get this neuron to this value. So let's see the code. So I have here the operation in reverse order, and if it's a convnet, I'm basically making this transpose convnet with weights initialized as the weights of the operation I have, and then I am removing so I'm basically subtracting the bias from the activation map and sending the result to this convnet transpose 2D. Pay attention, it doesn't have bias because I'm doing these operations reverse. At the same time, I'm computing the receptive field of that particular neuron in the operation we are performing right now. If it's a ReLU, we just do ReLU. It doesn't change the receptive field. If it's a max pooling operation, we use max unpooling operation with field computed properly. So if you have this receptive field from 2 to the 4 on this vertical axis, 2 to the 4 on the horizontal axis, after doing pooling, then the input should probably have this receptive field of 4 to the 7 without this 8s included. So if you pay attention that's the right one. No need to pay attention. So then after performing all the operations, I would receive this tensor of the input shape with receptive field computed correctly, and every pixel on that input tensor, constructed or reconstructed input tensor, has some certain values. I would basically crop that receptive field from the image and basically save it for the filter I, so that I can later visualize that patch for that so to say filter. So in short, what I'm doing is that I'm getting the activation map of the certain filter and choosing best 9 neurons in that activation map and trying to compute the receptive field of this neuron on the input image. So I'm not only computing the receptive field, I'm also computing the values of the pixels in that receptive field, so that the value of the neuron that I'm computing is exactly the same as we've discovered with the given input image. I'm also returning the receptive field area itself, so that we also can maybe just crop the input image in that receptive field and visualize that. So we have 2 types of visualization, just like in the paper here. Okay, so this is it. This is the class definition, and everything else is just like what you've done before. You know how to training. So I have convnet defined. I just move it to the CUDA, and I define certain hyperparameters. I'm doing cross entropy loss, and I have this in the test function and training function with these default values defined. Then I have this data set ready, and I would just do 20 passes on the image. It would reach approximately like 5 to 9% accuracy on the test set, which is kind of enough for us. Here I would stop the video and resume when the training is finished. Training has ended, and we reached 11% accuracy on the test set, which is slightly overfitting. We reached 9% and then started overfitting on the test set, but improved on the training set. It's okay. Optimization of the neural network is another kind of topic, and that requires another kind of videos. So we're basically going to work with training set images. Here I have this visualize function, which basically tries to select a random image from the training set and gets the patches of interest from case convnet, and then returns these patches so that we can visualize them using a plot image grid with borders function. So let's do this, and it would also return the index of the image we are working with, so that we can visualize that index too. We can visualize this image too. So now I'm visualizing the basically the last layer. Let's start with the first. So yeah, let's start with the first. As you can see, the first layer is basically trying to detect the certain patterns, and usually these patterns are either edges or vertical edges or the horizontal edges or certain edges of different angle, or they might be just colors. So here this kernel is detecting red color, and this kernel, and other kernels are detecting the vertical or horizontal edges and some other patterns that might be really interesting. So here we have this butterfly, which is really kind of dark. Let me actually send this index here too, index which is none at the beginning. So if int none, then generate it. If it's not, then we'll basically working with the value that we're interested in. So let's say we are working with 50s image, and the first pattern is basically this. Okay, and so this is at the image. If you remove this second variable, we can see that the patterns that are responsible for that are basically this. Okay, I'd repeat. So if you remember this, for example, this pattern, these horizontal edges, you can see that they really correspond to it. So the difference between two visualization is that the first one is what is required from the input image so that the particular neuron is activated, and what actually it is here given as the input image. Okay, we can now try to visualize the patterns in the second layer. Why not? And as you can see, the reception field is getting bigger, and then these patterns are getting interesting. So let's visualize the image itself. Well, this kind of an interesting butterfly. Let's visualize something else. What's there? Yeah, this should be a red butterfly. Yeah, this is a red butterfly. And then this is the patterns in the second layer of this conv net that are going to be highly activated, and they are detecting certain patterns, such as dots here that are really useful when detecting the family of the butterfly, and the colors obviously. So we can try to visualize the kernels in the third layer. So now the details are getting even looked at even from higher position. Okay, so and now our neural network start looking at this edges of the butterfly, this corner of the butterflies. So it's going to learn kind of what a certain family of the butterflies have, which kind of shape they have at this corners of their wings. Okay, then we can try to visualize the fourth convnet, which is in the second block. And now, as you can see, it's more about these patterns across the wings of the butterfly with some edges in it. And lastly, we can even try to visualize the sixth one, which surprisingly should be about the geometrical structure of the butterfly. As you can see, yes, it's more about how the geometrical structure of the butterfly is represented, and also what is the texture of the butterfly's wings, what kind of general shape it contains. And this is obviously basically the compositions of the patterns we've identified earlier. I guess this is a really good data set to play with if you want to visualize the filters of the neural networks. You could choose some other data sets too. You can find them in papers with code, but then this is kind of enough. if you want to see the patterns in the last layer, we can visualize it too. And they're going to be something like, yeah, it will take more time. Yeah, it's going to be something like that. Yeah. So these are the patterns. Obviously, this visualization kind of looks different because the colors are normalized, so they're not their true values. But then you can still see that there's a play between certain shapes. For example, here, you can see the triangle, or here this circular shape, or certain color conjunctions of more difficult complexity in this butterfly. We can switch off and just try to visualize the image at these positions. Yeah. And you can see that these are mostly about geometrical shape, geometrical structure of the butterfly to identify its family. Yeah, this is it. This was the exercise. I'll upload the code to a GitHub repo and share the link of the GitHub repo. You can find the link to the paper on the description of the video. Hit a star in GitHub repo if you like it. I'm open for contributions, pull requests, and you can write any concerns in GitHub issues.